Hello and welcome to my poster presentation. My name is Julia Kress. I'm a doctoral researcher working in the tumor biochemistry group of Professor Svenja Meyer-Johann at the Institute of Pathology in Würzburg. I'm very glad to have the opportunity to present my project with the topic NRF2 enables EGFR signaling in melanoma cells. Inside one melanoma tumor exist different reversible phenotypes which can be grouped into subtypes. In dedifferentiated melanoma cells, EGFR is highly expressed in a negative correlation with MITF. MITF is required for differentiation of melanocytes. Importantly, this phenotype, which shows low MITF activity but high EGFR, is characterized by invasiveness of tumor cells and drug resistances towards BRAF inhibition. We also saw that a high EGFR expression alone promotes a poor survival prognosis for patients with skin cutaneous melanomas, showing its potential therapeutic relevance. Such an aggressive phenotype can be driven by a stress stimuli. NRF2 is known to be the master regulator of the oxidative stress response, balancing pro- and antioxidants in the cell to ensure the redox homeostasis. What we already observed is that NRF2 represses MITF activity, thereby inhibiting the expressions of pigmentation antigens. After we saw a regulation of EGFR by NRF2 after siRNA, we addressed the question how NRF2 contributes to the pro-invasive and therapy-resistant EGFR high phenotype and by which mechanism NRF2 affects the transcriptional reprogramming and plasticity in this melanoma subpopulation, focusing on activation of EGF receptor signaling pathways. When we analyzed RNA sequencing and chromatin IP datasets, we observed that also the receptor ligands EGF and TGF-alpha were regulated by NRF2. Importantly, in the NRF2 chip sequencing data, we found a NRF2 binding site in the promoter region of EGF, which contains a typical antioxidant response element, which is also similar to classical NRF2 target genes. This chip sequencing was done in USCC62, a human BRAF mutated melanoma cell line with EGFR high, MITF low status. The experiments I will show in the following were also done in this cell line. Next to this, I performed an ELISA assay following a siRNA-mediated NRF2 knockdown. I observed a secretion of EGF into the cell medium, which was strongly reduced when NRF2 was silenced. In addition, EGF was stimulated by an H2O2 treatment, which I used to simulate oxidative stress. Interestingly, when we analyzed datasets of tumor entities with KEAP1 NRF2 mutated tumors compared to KEAP1 NRF2 wild-type tumors, we observed a significant elevation of EGF expression. Thus, not only oxidative stress, but also mutational activation of NRF2 induces EGF. For the EGF receptor and for TGF-alpha, we saw no binding site in the promoter region for NRF2, so EGFR and TGF-alpha could be regulated by an indirect manner, so that we used doxycycline-dependent MITF overexpressing cells. Indeed, TGF-alpha transcript levels were repressed as well as TGF-alpha secretion was reduced measured by an ELISA assay following the MITF activation. We did similar observations for the EGF receptor, where the MITF overexpression led to reduced EGFR protein and RNA levels. This was confirmed by linear regression analysis with TCGA data for skin cutaneous melanoma, where MITF mRNA correlates negatively with either TGF-alpha or EGFR mRNA. Furthermore, we observed that NRF2 can influence the receptor tyrosine kinase signature. NRF2 enables the expression of EGFR target genes when stimulated with EGF for different durations in control compared to NRF2 knockout cells. This indicates that NRF2 is important for an optimal receptor activation and the transcriptional signaling of EGFR. To summarize this, we showed that stress-inducible NRF2 binds directly in the EGF promoter, which causes enhanced secretion of EGF and positive stimulation of the receptor. NRF2 also blocks MITF activity, which results in a de-repression of EGFR and TGF-alpha. Thereby, EGFR is activated and NRF2 can promote therapy resistances, supporting the MITF low, EGFR high population, where EGFR and NRF2 can enhance each other in a positive feedback loop. I want to thank my group and all who are involved in my work. If you have any questions, I will be happy to answer them. Thank you.